Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is meant for 17th August 2020. After having celebrated Independence Day with lot of freedom, of course restricted freedom at your own home, let's move on to the next interesting chapter as well as interesting topic about DNA. As I said, if someone asks what is your secret, it is our DNA. So it's always advantageous to know about our own DNA and know what are its properties. So right from history of this, Professor Crick, I said, we are thankful to two professors, Professor Watson and Professor Crick, for having discovered double helical structure of DNA. Because of that, whatever research that is going on until today and in future also, it is because of the discovery of Watson and Crick. And there was a central dogma. What is the central dogma? People wanted to know how the information proceeds, what is the direction in which the genetic information flows. So it is always one-way flow from DNA to RNA, RNA to protein. So Professor Crick in 1958 proposed the central dogma of molecular biology. Central dogma is the unidirectional flow. Unidirectional means one direction flow of information from DNA to RNA and from RNA to polypeptide or protein. And there were two scientists, that is Temin and Baltimore, in 1970 introduced a concept of reverse central dogma. So central dogma is the unidirectional flow, DNA to protein. Reverse central dogma is the information of DNA is going to RNA and back. That is, RNA, from RNA information is coming to DNA. It is also called Teminism because it is discovered by Temin, hence Teminism, and it occurs in retroviruses. Retroviruses. So DNA undergoing transcription. I am using the word transcription. Keep this in your memory. We will be talking a lot about this transcription. DNA undergoes self-replication, DNA replication, and forms RNA. So this is transcription. RNA undergoes translation. Whatever codes it has got, that code language will be decoded and form proteins. That's called translation. So DNA to RNA, formation of RNA from DNA is transcription. Formation of proteins from RNA is translation. Now in reverse central dogma or Teminism, what happens is it is the RNA that forms DNA and vice versa. And from RNA, as usual, translation occurs and proteins will be formed. So the DNA will be in different forms in different organisms. Like in prokaryotes, they do not have definite nucleus, as all of you know. It is basically, it is nucleoid, something like a nucleus. So DNA packing in prokaryotes is carried out with the help of RNAs and non-histone basic proteins like polyamines. So there is no DNA. Prokaryotes do not have definite nucleus. The DNA is not scattered throughout the cell. It is held together with some proteins in a region called nucleoid. So there is no nucleus. Instead, in prokaryotes, nucleoid is there. What is that? Nucleoid contains DNA with some proteins. The DNA in nucleoid is organized in large loops held by proteins. What about in eukaryotes? Eu means clear, karyon means nucleus. Here, unlike prokaryotes, in eukaryotes, nucleus is very clear. In eukaryotes, DNA is stabilized with positively charged basic proteins called histones. Histones are positively charged due to rich basic amino acids like lysines and arginines. So histones are organized to form a unit of 8 molecules called histone octamer, as you are seeing in the diagram. And the octamers, you can see the nucleus and the histone proteins are encircled by DNA. That's nucleosome. So DNA packing in eukaryotes is carried out with the help of positively charged lysine and arginine rich basic proteins, and they are called histones. The unit of compaction is nucleosome. Histones are of 5 types: H1, H2A, H2B, H3 and H4. 4 of them, H2A, H2B, H3 and H4, are organized to form a unit of 8 molecules called as histone octamer, as you are seeing in the diagram. And the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. This is the structure of nucleosome. So a typical nucleosome contains 200 base pairs of DNA helix. And nucleosomes constitute the repeating unit of a structure in nucleus called chromatin. So this negatively charged DNA wrapped around positively charged histone octamer to form a structure called nucleosome. And the nucleosomes are seen as beads on a string, as you are seeing here. Beads, Rudraksha, beads on string structure under electron microscope. Nucleosome forms the repeating unit of a structure in nucleus called chromatin. So you can see the chromatin structure there. And in a typical nucleus, some regions of chromatin are loosely packed and stain light, and these are referred to as euchromatin, which is transcriptionally active chromatin. The chromatin that is more densely packed and stains dark is called heterochromatin, which is inactive. So you can see here chromatin. The chromatin is packaged to form chromatin fibers. These chromatin fibers further coiled and condensed at metaphase, during cell division, second phase, after prophase, metaphase. It forms chromosomes. So euchromatin, as I said just now, it's a nucleus, loosely coiled region, light stain, it's called euchromatin. Heterochromatin, on the other hand, it is dense and it is dark stained. So euchromatin is transcriptionally active than heterochromatin. Now, Frederick Griffith, as I was telling you, very interesting experiment he has conducted, and we must credit him with the great discovery that DNA is the genetic material. And he has given a clue, even though he has not named it as DNA, but thankful to him, he has given an idea what transpires from one to other. So Frederick Griffith conducted an experiment to show transforming principle, transforming from one to other, changing from one to other organism in bacteria. He conducted an experiment on mice and pneumonia bacteria called Streptococcus pneumoniae. You must remember the name of this biological name also, binomial nomenclature, Streptococcus pneumoniae. The bacteria are found as 2 strains. One is virulent and it is a smooth strain. The covering will be smooth. Another one is non-virulent but rough strain. So the smooth strain is abbreviated as S strain from now onwards. S strain bacteria produce a capsule and is pathogenic, means pathogenic, it causes disease. What disease? Pneumonia. R strain lacks capsule and is non-pathogenic. R strain is okay, it doesn't cause any disease. What is that Frederick Griffith has done? He has taken a rough strain first, non-virulent, given injection to the mouse, and the mouse survives, as you are seeing here. Mouse lives. Now he takes a virulent one, rough strain. That is, virulent is a smooth strain. He gives injection. Look at that. The mouse dies. So that means there is the virulent smooth strain is very dangerous one. And then, interestingly, what he has done, he has taken the smooth strain and he heated it. And after heating, he has injected to the mouse. And the heat killed smooth strain did not do any influence on the life of the rat. Rat survives or mouse survives. Now, interesting one, fourth one. A rough strain which is not pathogenic. Remember, first injection, rough strain and heat killed smooth strain. Now he has mixed up. And when he injected this, what happens? Look at that. Unfortunately, the mouse dies. That means, as such, the rough strain is not dangerous. But rough strain is becoming bad because of the friendship with heat kill. That's why we say, your parents will tell you always, savas dosha. So, rough strain, which is good basically, which does not kill rat, in the company of bad fellow. Who is bad fellow? Heat killed smooth strain has become bad and kills the rat. So this gave an idea to Frederick Griffith. What is that? When R strains are injected into the mouse, it is non-pathogenic and does not cause pneumonia to rat. So the mouse continued to live. When the S strains are injected into the mouse, that caused pneumonia and mouse died. Now, when heat killed S strain, what he has done? He has taken the S strain pneumonia bacteria, Streptococcus, and heated it and injected into the mouse. That does not cause pneumonia. The mouse continued to live. But the dangerous combination is when the heat killed S strain mixed with R strain and injected into the mouse, pneumonia stuck to the mouse and the mouse died. What is the conclusion? This is interesting. Conclusion of the experiment. R strain bacteria had been transformed by the heat killed S strain bacteria. You see, basically R strain doesn't kill the mouse because it doesn't cause pneumonia. But R strain in combination with S strain, that too heat killed S strain, that's why people say habits die hard. Remember, heat killed S strain bacteria kills the mouse. The transformation of R strain, who is basically good boy, to S strain, bad boy, is due to transfer of genetic material. But Griffith was not knowing what is that genetic material. But he has proved that there is something which is getting transferred from S strain to R strain. The biochemical nature of genetic material was not defined from his experiment at that time. But we continue to explore many more experiments